Today I'll be reading an article by Peat. This is on his website, raypeat.com, and it's in the article section, and it is called Cataracts, Water, Energy, Light, and Aging. This is how it goes. Because of the baby boom population bulge, the market for cataract surgery and the little plastic intraocular lenses is growing wonderfully. According to the World Health Organization, there were about 20 million cataract surgeries performed in 2010, with 32 million expected in 2020. In the US, about 3 million cataract surgeries were performed annually. Revenue from sale of the intraocular lenses in the US alone was $775 million. That's with an M, and that was in 2010. And is expected to reach $965 million by 2017. In 2010, the Alcon company earned $1.2 billion with a B dollars from one type of intraocular lens, and that's from marketresearch.com. To promote the sale of the quote-unquote premium lenses, which cost thousands of dollars, patients are told that the more expensive lenses will save them money in the long run by making ordinary glasses unnecessary. And in parentheses we have sometimes. The lens replacement surgery is now sometimes recommended when a cataract has caused only a slight decrease in visual acuity, or even a suspected decrease in acuity. I haven't known anyone who had the surgery who had been informed of the incidence of complications of the surgery, which result in permanent blindness for thousands of the patients every year. Some of the causes of cataracts have been known for many years, but the knowledge is usually ignored by the medical profession. Medical myths about the causes of disease support present practices. Myths about the causes of cancer, heart failure, hypertension, menopause, osteoporosis, sarcopenia, depression, dementia, and cataracts are designed to reinforce each other, forming an interlocking system, an ideology of the organism. The conventional ideology identifies pathological cells and defective proteins and bad genes as the causes of organ failure and disease, and quote-unquote aging is seen as a dimension in which entropy tends to increase those defects. This ideology discourages thoughts of quote-unquote field effects in which the function of a molecule, a cell, or an organ affects and is affected by things that aren't in direct contact with it. This is why the removal of a lens is treated so casually. There is some knowledge about the effects of systemic disease on the eye, but very little about the effects of particular parts of the eye on systemic physiology, and relatively few physicians are aware of the effects of one part of the eye on the other parts of the eye. A few of these physiological interactions within the eye are very interesting. For example, injury to the lens powerfully stimulates regeneration of nerves in the retina. And that's from Fisher et al. 2000. Things which injure the lens enough to cause cataracts to develop might also be injuring the retina, but the emission of stimulating substances from the lens must be a compensating influence. Every normal tissue of the eye is emitting substances that affect other parts of the eye, and probably other parts of the body. Until the 1970s, the literature was dominated by the view that the lens was a lifeless material, like hair and toenails. And even in 2013, there is great reluctance of researchers to recognize its vital cellular activity. After an artificial lens has been implanted, there are great changes in the vitreous humor, which fills the space between the retina and the lens, with a reversal of the gradient of viscosity, and with changes in many proteins, including transthyretin, alpha-antitrypsin, retinoic acid binding protein, antioxidant proteins, and the enzymes carbonic anhydrase and triosphosphate isomerase. I think that's spelled incorrectly there with an extra ER. Anyway, that's from Neal et al. 2005. I haven't seen any recent studies of the effects of lens removal on the nervous system, but a 1953 study of 21 patients reported a high percentage of behavioral disturbances following the surgery. This is a quote. Following the operation, 20 patients showed some alteration in behavior, including changes in mood, psychomotor disturbances, paranoid and somatic delusions, hallucinations, disorientation, and confabulations. In 3 cases, the disturbance was characterized as severe. Here's another quote. It is concluded that disturbed behavior is an integral part of the reaction of almost all cataract patients because of a complex interaction of a number of factors. And that's from Lin et al. 1953. In animal studies, when the lens capsule is closed after removal of the lens, within a few weeks, a well-formed lens has regenerated. That's from Guan et al. 1993. Cell division is stimulated in the cells remaining attached to the capsule, similar to the regeneration of the adrenal cortex after its removal. Artificial replacement lenses are designed with an ultra-sharp edge to block the regenerative migration of cells within the capsule, because the cells can quickly form a new cataract behind the plastic lens. Those cataracts commonly form in reaction to the lens. The use of arsenic to kill these cells has been proposed, and probably used. And that's from Zhang et al. 2010. The easy money in lens surgery has obviously discouraged professional interest in preventing cataracts, or curing them, or stimulating the regeneration of new lenses. Research in the prevention of cataracts has encountered serious barriers to performing the clinical trials that would be necessary for approval. Here's a quote. Clinicians have even developed the opinion that lens and cataract research is no longer necessary to overcome cataract blindness. That's from Sasaki et al. 2000. However, it isn't inconceivable that someone could find a way to make prevention, cure, or regeneration significantly remunerative. Although the lens has no blood supply, fluid carrying nutrients and oxygen is constantly flowing through it, providing the cells with glucose, amino acids, and ATP that it uses for maintaining its structure. Its proteins are being renewed continually, broken down, and synthesized. It's from Ozaki et al. 1985. There is clear evidence that some of the core cells retain a nucleus, and that large molecules can move between cells. That's from Lieska et al. 1992, and Shestblov and Basnet 2000, Stewart 2008, Matthias and Ray 2004. Despite this evidence, prominent researchers are still promoting the paradigm of inertness, the lens as analogous to a toenail. As in other cells, ATP maintains the proper water content in the cells. Besides providing energy and amino acids, the circulating fluid carries minerals and many hormones and regulatory substances. The absence of a blood supply to the lens has kept people from thinking of its pathology in terms of the inflammatory processes that are now recognized in other conditions, for example in dementia, heart disease, and cancer. But the same basic processes can be seen in the development of cataracts. Improved knowledge of lens physiology is very likely to lead to major improvements in therapies for the other conditions. In the lens, the state of water changes before there is any other evidence that a cataract is developing. That's from Mori, 1993. Detecting similar water changes in other tissues might improve diagnosis and treatment of other problems. Things that actually lower the ATP content of cells increase their water content, and in the process, the water functions differently, becoming more randomly arranged. The idea that the properties of water change as cell functions change contradicts the common reductionist assumption that water is just the medium in which molecular interactions occur. Since Kelvin's 1858 demonstration that the heat capacity of water changes with its shape, The Drost-Hanson's demonstrations that its density decreases near surfaces. Attention to the physical properties of water has made it possible to understand many biological mysteries, such as the decrease of volume, Abbott and Baskin, 1962, when a nerve or muscle cell is excited. Although the invention of the MRI grew directly from Damadian's understanding of water's centrality to biology's most important issues, the technology's most important contributions related to changes in water structure haven't been recognized, understood, or assimilated by medicine. The electrical properties of the protein framework of a cell interact with the state of the water in the cell, and with the things dissolved in the water, including phosphate, calcium, sodium, and potassium. Actin, one of the major muscle proteins, forms a meshwork in the cytoplasm of lens fiber cells, and myosin, the other major muscle protein, has been found in association with the actin. That's from Al Ghul et al. 2010. ATP, alternating with ADP plus inorganic phosphate, is involved in muscle contraction and relaxation, and it is involved in the conversion of actin from a filament into a globular form. Changes in the amount of ATP and ADP are important for influencing the interactions of water and proteins. The actin skeleton is involved in the fiber cell's elongation as it develops from a roundish epithelial cell, and it's probably responsible for the ability of lens cells to contract when stimulated. Opitz et al. 2003 and Angelica et al. 2011. These muscle-like effects of actin are believed to be responsible for the movement of organelles and other cell motion, such as cytoplasmic streaming. But, as a major part of the cell's structure, it could also be expected to act as the framework for electroosmotic flow of water, accounting for the circulation that maintains the cell's energy. The observed static electrical properties of lens cell fragments could account for a complete daily renewal of the fluid, Pasquale et al. 1990. But the metabolic gradients in whole cells would probably cause faster flow. With oxidative energy production occurring in the surface cells, an electrical gradient will be created, causing water to flow away from the site of respiration. Electroosmosis probably also accounts for the somewhat mysterious exit of water from the eyeball and brain in perivascular flow. The flow of water through these cells is very fast, but Ichiji Tasaki has demonstrated similarly fast movement of water in nerves and artificial polymers in association with electrical activity, 2002. Also, Tasaki and Iwasa in 1981 and 1982, and Iwasa et al. In 1980. At least since Gullstrand's unfounded assertions in his 1911 Nobel lecture, it has been assumed that the lens, like a water-filled balloon, keeps the same volume when it flattens. For distant focus, Zamudio et al. 2008 have shown that, and this is in quotes, the lens volume decreases as the lens flattens during unaccommodation. And another quote, the lens volume always decreases as the lens flattens. They determined that, another quote, the changes in lens volume are reflected by the speed of the equatorial diameter recovery in vitro cow and rabbit lenses during simulated accommodation, occurred within a physiologically relevant time frame, 200 milliseconds, implying a rapid movement of fluid to and from the lens during accommodation. This is the duration of the action potential of healthy heart muscle, although it's probably not as fast as the very superficial changes that Tasaki saw in nerves. It's the sort of change rate that could be expected in an organ whose change of shape is the result of stimulation. Accommodation, with this immediate hydration, is produced by cholinergic stimulation, and in the healthy lens this hydration is rapidly reversible as the stimulating acetylcholine disappears and the lens flattens. The failing heart muscle, unable to relax fully, becomes harder as its water content increases, and cancer cells, locked into a contracted, excited state, become stiffer as their water content increases. Similarly, cataracts have been described as more rigid than normal lens tissue. Hayes and Truscott, 2008, and Hugh et al., 2000. Yet their water content is higher, and that's from Rex et al., 2000. Along with the increased water, the stressed cells take up very large amounts of calcium, and sodium increases while potassium decreases. Inorganic phosphate increases in the stressed cells, some of it entering with the circulating fluid, but some of it produced from the ATP, which is decreasing. Serotonin, iron, lipid peroxidation products, nitric oxide, and prostaglandin are also increased. The increased calcium activates proteolytic enzymes that break down protein. In the failing heart and growing tumors, there is an increase in the quantity and the cross-linking of collagen in the extracellular matrix, contributing to the overall hardness, besides the contracted state of the cells themselves. In the cataract, cross-linking of various proteins, including collagen, also seems to be involved in the problem, along with the altered state of the water. That's from Mishra et al. 1997 and Eldred et al. 2011. The cross-linking enzyme transglutaminase is induced by stressors such as ultraviolet light, which produces cataracts. When the available energy doesn't meet the cell's energy requirements, if the cell isn't quickly killed by the stress, it will use some adaptive mechanisms, stopping some repair processes to reduce energy expenditure, possibly stopping specialized functions to reduce energy needs. Fibrotic changes occur as a result of defensive reactions in stressed cells, usually following long periods of fatigue and inflammation. Cortisol generally protects cells by blocking overstimulation and providing increased material for energy and repair. But it can kill cells, nerve cells and thymus cells, that depend on glucose oxidation, leading to immunodeficiency and excitotoxic brain damage. The glucose-dependent lens fiber cells express the same glucose transporters, GLUT1 and GLUT3, as the brain, and the quote-unquote nerve-specific GLUT3 is concentrated in the dense nucleus of the lens. That's from Donaldson et al. 2003. Exposure to excessive cortisol or hypoglycemia is able to quickly produce cataracts, showing the basic importance of glucose metabolism for lens health. Oxidative metabolism in the surface cells is probably largely responsible for the streaming of fluid through the fiber cells, providing some ATP and the nutrients that allow the fiber cells to maintain and repair their structure. But I suspect that local metabolism of glucose by the fiber cells provides most of the energy for keeping the protein-water system in its orderly, relaxed state. The aging lens, like all normal tissues, is drier, has a lower water content than younger tissues, but when a cataract begins to develop, there is a sharp increase in the water content in that area, something that happens in any excited or fatigued tissue. In a stimulated nerve or muscle, for example, although in a closed system there would be a slight decrease in volume as its water becomes relatively randomized, there is normally a sudden absorption of water from the extracellular space, where the water has the same random organization. With the decreasing energy charge of the cell represented by decreasing ATP and increasing ADP and inorganic phosphate, the long-range order of the water decreases, changing the activity of enzymes in a variety of ways, for example, by the change of a high magnesium content for a high calcium content, while the renewal of proteins decreased because of an energy deficit. The activation of proteolytic enzymes by calcium degrades the cell architecture and the crystalline that makes up 90% of the cell's protein, and these damaged proteins become progressively cross-linked in a process analogous to the cross-linking of collagen in sun-damaged skin, or in cancer or fibrotic failing heart, or a fibrotic failing heart. The diffusion of water in these congested cataract areas becomes random, more like ordinary bulk water, and it's likely that this randomization of the water, along with the architectural disorganization of proteins and changing electrical fields, impedes the longitudinal flow of nourishing fluid through the lens. MRI studies show relatively free diffusion of water longitudinally in the lens fiber cells from front to back, but not transversely, and that's from Moffitt and Pope, 2002. Water that's highly ordered by nearby surfaces can still be very mobile parallel to the surface. The parasympathetic nerve transmitter acetylcholine is formed in the lens, as well as its receptor and the enzyme which destroys it. Cholinesterase, not a word I say usually. Chemicals that inhibit cholinesterase and drugs that mimic the action of acetylcholine on the receptor cause cataracts. These drugs, and this is from Maikon and Kinoshita, 1968, and Harkonnen and Tarkkanen, 1976. Again, these drugs cause the lens to take up water, sodium, and calcium, and to lose potassium. And by increasing the cell's energy expenditure, they accelerate the consumption of glucose while blocking other metabolism. Since these are known effects of stimulation by acetylcholine, it's reasonable to assume that acetylcholine is involved in the natural formation of cataracts. Besides the direct excitatory effects of acetylcholine, the increase of intracellular calcium and decrease of magnesium from Agarwal et al. 2012 caused by it promote the synthesis of nitric oxide, which, for example, blocks the function of cytochrome oxidase, reducing the production of ATP, and the interference with glucose metabolism in itself is cataractogenic, causing cataracts. And that's from Grainer et al. 1981. Cataractogenic. There it is. Ultraviolet light powerfully stimulates the formation of nitric oxide. This is from Chandri et al. 1993. And it is one of the known causes of cataracts, since the cornea is more directly exposed than the lens to the ultraviolet rays of sunlight. The effects of injury can be seen more quickly. Exposure of the cornea to ultraviolet light causes swelling, reduced transparency, and the formation of nitric oxide, which enters the aqueous humor. That's from Sejka et al. 2012 and Sejkova et al. 2005. Swelling in itself, regardless of the cause, decreases the transparency of the cornea. That's from Stevenson et al. 1983. Anything interfering with its energy metabolism causes swelling. The blue color of ordinary water is caused by its absorption of red light, possibly by its hydrogen bonds. That's from Braun and Smirnov, 1993. But there haven't been many studies of the physical effects of red light on water itself, since water absorbs much more strongly in the infrared wavelengths. There is a tendency to explain the benefits of sunlight by its infrared rays. Red and orange wavelengths penetrate tissue very effectively because of their weaker absorption by water, allowing them to react with pigments in the cell, such as cytochrome oxidase, which is activated or reactivated by red light, increasing the production of ATP. This effect counteracts the toxic effects of ultraviolet light, but there are probably other mechanisms involved in the many beneficial effects of red light. Recent work by a group at the University of Ulm in Germany, Andre Sommer et al. 2011, has revealed an effect of red light at 670 nanometers on water that I think helps to explain some of its protective and restorative actions. Shining laser light onto layers of water adsorbed on a solid surface, they were able to show, quote-unquote, a breathing-like volume expansion of the topmost sheets of water molecules. They explain this as the result of a stabilization of a more ordered state of the hydrogen bonds of the water. They are applying to this to chemotherapy, since the expansion of water in the cell, where much of the water is in adsorbed layers, similar to their experimental setup, alternating with its volume contraction as the light is pulsed, causes water to move in and out of the cell quickly, taking some of the drug with it. They have also proposed that degenerative changes in the connective tissues involve a loss of ordered water, and have experimented with light treatments to restore elasticity and flexibility. Since the water in cataracts is in a less ordered state than in the transparent lens, the reordering effect of red light could be valuable, and if the effects are the same as in their experiments with cancer cells, the increased volume of the reordered water would cause a movement of water out of the cataract as it does in cancer cells in their experiment. And the known restorative effect of red light on oxidative production of ATP would almost certainly be helpful. Among the popular medical treatments that are likely to contribute to the development of cataract are glucocorticoids and drugs that increase serotonin. That's from Dietz and Tilgner, 1973, and Korsakova and Sergeva, 2010. And drugs that increase nitric oxide. Free fatty acids are toxic to the lens, which contains the enzymes for synthesizing prostaglandins and related promoters of inflammation. The products of lipid peroxidation are increased in people with cataracts. Endotoxin from the intestine increases the formation of nitric oxide, so it's essential to minimize intestinal inflammation. High altitude very strongly protects against cataracts. That's from Brilliant et al. 1983. Low oxygen tension itself protects the lens's clarity, from Akoyev et al. 2009, possibly by the protective effect of increased carbon dioxide against glycation of protein amino groups. Aspirin's known anti-cataract effect apparently involves a similar protection of crystalline against glycation. But aspirin has several other protective effects, including prevention of protein cross-linking and the inhibition of the synthesis of nitric oxide and prostaglandins, and other disruptive materials. That's from Crabb, 1998, Beachy et al. 1987, and Longchamp et al. 1983. Progesterone's inhibition of nitric oxide production is probably protective for the lens, paralleling its effects in other organs. Inhibitors of nitric oxide, such as aminoguanidine, are protective. Cholinergics, including atropine, inhibit overhydration of the lens and prevent cataracts caused by excessive cholinergic stimulation. That's from Kaufman et al. 1977. Caffeine in animal experiments prevents cataracts. Uric acid, which inhibits nitric oxide formation, is reduced in people with cataracts. The factors that prevent or promote other degenerative diseases are similarly protective or harmful for the lens. And once again, he's got all his references. So if that's you wanting to go through all of these, great. But again, as usual, Peat has done a great job of relaying this information to us in a way that I don't think anyone else has been or is able to do. And so again, I'm very much appreciative of his work, and I will see you all next time.